In this short video, we will talk about the microRNAs. MicroRNAs are the class of non-coding RNAs that play an important role in regulating gene expression. So microRNAs are getting involved in the gene regulation. So microRNAs are associated with the development of an organ, let's say brain. It is involved in the cancer. It is also involved in regulation of large gene networks. We will talk about all of this in this video. So first let's see how microRNAs work. So these are dedicated genes in our genome for the microRNAs. So these gene doesn't encode a protein, but they encode a microRNA. So the first transcript that comes out of these kind of genes are known as primary microRNA transcript. And these primary microRNA transcript get further processed with the help of enzyme known as DROSHA and DGCR8 or PASHA. So the pre-microRNA transcript gets further trimmed. Eventually, this particular pre-microRNA transcript would be moving out of the nucleus with the help of the exporting 5 complex as shown here. Once outside in the cytoplasm, it would be assembled with several other proteins known as dicer, and dicer further chops off this kind of loop structure at the end. And now there are 2 different strands of these microRNA which would eventually be associated with arginate protein, and it will eventually form the RISC complex or RNA. Silencing complex, only one strand of these microRNA would be loaded into RNA silencing complex. It binds to the specific region of mRNA, and in most of the cases it leads to the degradation of mRNA. This is how overall microRNA can work, but there are many aspects to it, like the production of microorganisms would be way more complicated that we have seen right now. So at a simplest level, there would be one microRNA from one particular microRNA gene. There could be also multiple microRNA or an array of microRNA coming out of from one gene, and later on they are getting processed. There could be also microRNA as a part of an intron in a protein coding gene, so all these possibilities are there in a live cell. So the question is, where does microRNAs bind? MicroRNA can possibly bind to different untranslated regions. Most commonly, they prefer 3' untranslated region. And some of the cases, though rare, they can also bind to the 5' untranslated region, but that's much less frequent. So how does microRNA regulate gene expression then? MicroRNA can regulate gene expression in 2 ways. First of all, it can downregulate the mRNA by degrading it just by associated with the silencing complex. It can chop off the mRNA and thereby less mRNA would be available to produce protein, so less product would be formed. There is also another way by which microRNA can work, that is interfering with the translation. Some of the cases the microRNA and the RISC complex sites sits in a 3' UTR, let's say, but does not cleave the mRNA. So it works like a roadblock for the ribosome. It interferes with the translation process. That is also possible. Now let me tell you, the microRNAs are also present in a tissue-specific and cell-specific manner. So let's say there are different cell types like epithelial cell. One microRNA might not be present in this cell but might be present in neuron, and so might not be present in fibroblast again. So there could be specific microRNA which are very cell type specific, but there could also be microRNA which are present in pretty much all the cell types. So depending upon these functions and availability, microRNA regulate the overall gene regulatory neurons in different cell types. So how does that actually happens? We will imagine that from a stem cell we have 2 alternate possibilities, one epithelial cell and one neuron. So there are 2 master regulatory network here shown by master regulator 1 and 2. So these master regulators are transcription factor, and these regulates one particular gene expression module, and each of these molecules actually suppress each other. That's why in the stem cell state, none of the fate is acquired. So one of the module has to dominate over the other module. That is how the system is laid off. Imagine a microRNA which is, let's say, microRNA X. This can suppress the master regulator network 2. So as a result, the master regulator transcription factor would be produced less. The gene module would be poorly regulated or downly regulated. So now the repression of the module 2 over module 1 would be gone. And now module 1 would be the predominant gene expression module mediated by master regulator 1. So it might lead to the fate towards an epithelial cell. So this is actually how we saw how one possible way by which microRNA can regulate the cell fate choice. MicroRNA are not only present in cell but also present in secreted body fluids. For example, microRNAs can be found in cerebrospinal fluid. One example is microRNA 204. It has been shown by the scientists recently that the microRNA 204 regulates the quiescence of stem cells in the brain region. So it regulates the number of neurospinal stem cells, and that is super important from brain development. MicroRNA are also implicated in the context of cancer. Small RNA sequencing from the tumor samples has identified that there are several microRNAs which get upgraded in a cancer sample. So, how does they regulate proliferation of or cell division? Kinetics is something to note in future years, but microRNAs are super important in context of many diseases, including cancer. So let's ask that how can we study microRNA experimentally? So, microRNA can be detected with the help of small RNA sequencing, but that's very much expensive. So, if we have to know about very few microRNA, let's say 4 or 5 microRNA, quantitative real time PCR would be another alternative and very sensitive method. Also, fluorescence in situ hybridization can show you the intrinsic localization of a particular microRNA inside the cell. There are reporter assets as well. In this reported assay, there could be a reported gene like GFP. Underneath the reporter, the 3' UTR of a target mRNA would be provided. Anyway, this reporter would be eventually transcribed and translated to give rise to the GTP. But imagine that there is a microRNA which binds to the 3' UTR and degrades this particular mRNA. So, in that content, the less amount of GFP would be produced. So, by looking at the GFP, the fluorescence one can get an idea that the regulation of microRNA for this particular transcript, this show that one can possibly study the microRNA in the lab. So I hope that you liked the video and it was very useful for you. Thank you so much.